Welcome back, everybody. This is part 1B. So in our last part, we discussed some of the main concepts of chapter 2 and chapter 3. And let's go ahead and take a look at what chapter 4 is going to bring us. We're going to take a look at some sex determination and sex chromosomes. Alrighty, so first things first, when we take a look at sex determination when it comes to humans, we're going to go ahead and from a genetic standpoint basically take a look at our sex chromosomes, which are our X and our Y. And we will also discuss in this chapter the fact that you don't always need to rely on those particular chromosomes. There are some variations, and there are also some other mechanisms that can exist in determining the overall sex, including environmental as well as some behavioral attributes that will come into play. All right, now for Homo sapiens, we are utilizing our XY sex determination, and it basically says that when we look at a genetic male, we are looking at the inheritance of an X and a Y chromosome. We also like to describe the combination of both of the letters as an heterogametic sex because hetero means one of each. So as you can see that you would need an X and a Y. One parent would have to contribute the X, the other would have to do the Y for it to be a genetic male. Keep in mind that in addition to the sex chromosomes, you also inherit your 22 pairs of autosomes, which are the same between the sexes. From a female aspect, from mammals and from humans, what we see is that you would need to inherit two X's. So you are a homogametic sex, meaning that both parents would have to give you the X for you to be a genetic female. So if you think about it, if males are X and Y and females are XX, then the parent who determines the sex of the offspring is going to be the father. I hope you said that. And in case you're not really sure, go ahead and take a look at the little picture right over here. And then notice XY is for the male, two X's is for the female. And remember our law of segregation basically says that when it's time for the gametes to form, the guy will either give an X or a Y. And ladies, we will only be giving our X's. So that means that the mom always donates the X, and the father will either donate a second X, making a female, or will donate the Y, making the male offspring. Now, there are lots of variations out there, so we're only going to talk about a few of them, but these are the most popular ones, and I think they're also the more interesting ones. So, for instance, if we were talking about mammals and we were talking about humans, the X and the Y, we get that, all right? We're familiar with that, but what if we start looking at different insects? So, for instance, grasshoppers, we see that what determines if it's an X or if it's a male or female is basically going to be the presence of how many X chromosomes. So in this case, we have what we like to call an XO sex determination. And the O basically means that there's nothing there. It's absent. So from a grasshopper standpoint, and this actually happens in a lot of insects, we see that if the male only inherits one X chromosome, then it's genetically a male. I'm sorry, if the offspring only inherits one X chromosome, it's genetically a male. A female will inherit both copies. So you'll have XX for the females. So it's all about the ratio of X chromosomes compared to autosomes. So at the bottom of the slide, it says that if you only get one sex chromosome, one X chromosome, compared to your normal number of autosomes, then we're looking at a male. So this is our X and nothing else. That's what the O represents. Where on the other hand, if you inherit two of your X chromosomes and you put that over the normal amount of autosomes that you will inherit, then we're looking at a genetic female. You don't always have to rely on the X and the Y chromosome. There are chromosomes that look distinctively different in different animal species that will have a sex determination role, and that's the case when we look at birds and fish. Instead of an X and a Y, we use a Z and a W because the chromosome looks different. And what we see happening is that if you're looking at a male, you're looking at a homogametic sex of inheriting 2 Z's. Okay, and if you're looking at a female, now we have a heterogametic sex because we're looking at the interplay of 1 Z and 1 W. So isn't that cool? So we have 2 different chromosomes mixing, and in this case we have the male who's homogametic because it's taking 2 Z's, and the female who's heterogametic, she's taking 1 of each. Another scenario that you come into that's actually been studied quite extensively in bees and to some point in ants as well is what they like to call the haplodiploid sex determination. And the haplodiploid takes a look at the complete number of sets of chromosomes. So are you going to inherit only the haploid number, or are you going to inherit the diploid number of chromosomes that are available? And like I said to you, this one's been heavily studied in bees. So the example we're going to look at is the honeybee. So what we see happening is the male species in honeybees, aka a drone, is the end product of an unfertilized egg. And this unfertilized egg will only have 1 set of chromosomes, so only the maternal contribution because it's unfertilized. So we're looking at a haploid organism. And from a sex determination, that will then allow the male to be distinctive. If we're looking at an egg that becomes fertilized, meaning that it now has a diploid copy, so 2 contributes, that what we see happening is that the diploid amount of chromosomes will automatically give rise to the distinction of the female sex. So when it comes to honeybees, the females are the only ones that are being produced as an end product of sexual reproduction. The males are haploid because they originate from an unfertilized egg, whereas the females are diploid because they come from 2 sets of genetic information due to the fertilized eggs and obviously having their diploid number. And I'm sure living in Florida, you've heard about the alligator and the fact that the temperature can determine its sex. So what we see happening here is an example of environmental factors that can contribute to the overall phenotype and development of the offspring. And we see this in some reptiles as well as in some fish species. So the example that we stuck with is for the American alligator. And studies have shown that if the eggs are incubated at around 33 degrees Celsius, they're going to grow into males. If we start varying, either if we go above or below that 33 degrees Celsius, we see that they will go ahead and grow into females. They have all the same chromosomes composition, so it's not a difference in diploid versus haploid. It is all about the expression or the variation in gene expression that will determine the male versus the female sex. Another really nice environmental feature, this is really more of a behavioral feature that I want to discuss with you, is that of the clownfish. And every time I give this example, I keep thinking about, that little cartoon, Finding Nemo. And, there's something wrong with the cartoon, but maybe as a biologist, you don't want to think about it because, we enjoy watching the cartoon. But anyways, I discussed this, I talk too much. So let me give you my little example. So in clownfish, what we see is that they are hermaphrodites. And they have the ability to basically switch from male to female. And usually what we see is that within a little colony, you will have 1 female, you will have 1 male, and then you'll have a lot of small juveniles. And the juveniles usually have both of the immature sex organs, so they can go either way. And what we see happening is the structure of the colony is going to be that if something were to happen to the female, the male will go ahead and, let's say the female dies off, excuse me, I should say it. So let's say the female dies off. What would happen is that the male will go ahead and, utilizing its hermaphrodite properties, will metamorphosize and go ahead and take over the female role. So the male will switch over and become the females. And within the juvenile population, the largest one will then go ahead and become the male fish. So the family unit will be rebuilt with the male becoming the female and the biggest of the juvenile becoming the male role. Now, I should tell you that usually the female clownfish is larger than the male clownfish. And what we see happening is that the female tends to control not only the male, but also the juvenile by very aggressive behavior. And studies have shown that the way that the male and the juveniles react to this aggression that the female is exerting is that somewhere or another the brain basically goes ahead and suppresses the amount of estradiol that's being secreted in the males and the juveniles, allowing them to stay either as the male or in their immature state. And it's only when you go ahead and you remove that aggressive force from the female, so when the female dies, that we see that the male is able to excrete higher levels of estradiol, and thereby the male can go ahead and metamorphosize into the female. And then the juvenile, because there's no aggression, no suppression, the largest one will then go ahead and be upgraded to the male. In an aquarium, usually what we see happening is that if they are just left by themselves in an aquarium with no aggression surrounding, the default sex does tend to be a female. However, if you start combining them together, then usually the largest one will stay the female, and because of the behavioral attributes and the high levels of aggression, we see that the smaller one will go ahead and will be dictated to as the male. So bringing this back to our Finding Nemo theory, that means that if Nemo's mom died, then Nemo's dad should have become his mom. You see how it just complicates the whole story? All right, I'll stop talking. But either way, I always thought this was kind of cool because they are hermaphrodites. So they have the ability to switch from males to females. And basically what keeps them in check is the aggression of the largest clownfish, which will automatically be the female, suppressing the estradiol secretion in the smaller units of her colony. Okay, so another thing that I want to kind of bring up is what we like to call dosage compensation. And this really plays a role when we start looking at our X chromosome. So I should have put a little picture of our karyotype in here. But in our previous lecture, I showed you a little karyotype where you had your 46 chromosomes paired up and kind of lined up. And I showed you, this is what the X looks like, this is what the Y looks like. And you might have seen, or you might have remembered, that the X chromosome is significantly larger than the Y. So we basically see that there are a lot more genes on the X chromosomes than there are on the Y chromosomes. And we'll talk more about the size difference in a little bit. But what we see happening is, obviously, we want to make sure that if you have a male and a female species, that their dosage of X chromosome representation needs to be kept in balance for both, regardless if we're looking at having a duplicate or just one copy of the X chromosome. So dosage compensation basically takes a look at what mechanisms are we utilizing to control the expression of the X chromosome across both sexes. And there are different ways to kind of approach this, right? So if we're looking at organisms that use the X and Y expression, we can see that if you're looking at a male that's X and Y and a female that's XX, you can do it two ways. Since the female has two X's, that means they technically have twice as much of the dosage of the gene. So how do we control that doubling versus just the one X that we see in males? Well, we can either increase the expression in heterogametic options. So if we see that you have an XY option, you would increase the expression of the X, you would amplify it up to the level of the species that will have two X's. Or you can decrease the expression, meaning that if you're looking at a homogametic sex, so a species that has two X's, such as the females in mammals, we would decrease the expression by obviously silencing one of the X's. so that across the board in both the males and the females, they would only have one representation of one X chromosomes. And that's actually the popular choice when it comes to mammals. We tend to decrease our expression of the double X's in females, and we're going to do this by something called X chromosome inactivation. And I want to kind of talk to you a little bit about how that's done and how it gives us what we call bar bodies. Go ahead and take a look at table 4.1. It gives us different examples of species that we've come across in the XX and the XY variation, and it talks to you about the different mechanisms of compensations. So you're going to notice that oftentimes you can either look for a way that you can increase the expression of the X chromosome in the male species, or you're going to look for a way that you can diminish the expression of one of the X chromosomes in the female species. Your end goal with this whole dosage compensation is that you figure out a way that the genes for the X chromosomes are equal in both male and females. All right, so as I mentioned to you, mammals like to do X chromosome inactivation, and that basically means that very early on in development, randomly in all of the cells, one of the X chromosomes is going to become inactive. And it's going to become inactive by what we like to call the formation of a bar body. A bar body is a staining process that basically shows us when the chromosomes become condensed, that it coils up and it will stain darker when we make our histology slides. And I'll go through the actual process of what creates a bar body in just a few slides, but for now I just kind of want to show you this on the illustration. So let's take a look at the picture that's at the bottom left-hand side. And we have a close-up of the nucleus. And what we see happening right here in this light spot right here, this is what we call a bar body. And this is basically one of the X chromosomes that have been coiled up and they have been condensed, they have been inactivated. And then when they're applied to the stain, it's able to take up the stain in a more condensed factor because of the coiled up pattern of it. So it will kind of shine brighter. It might be better to take a look at the illustration next to it, where you can see here in yellow, I'm going to put a little circle around it, that is our activated X chromosome. And then right over here is our bar body. This is our inactive X chromosome. And this X chromosome inactivation, it's all based on the line hypothesis, basically says that very early on in development, randomly in each of our somatic cells, we're just going to deactivate one of our X chromosomes. So there isn't a pattern. It will just go ahead and it will activate one here, one there, in every single one of our cells. And it doesn't pick only the maternal or the paternal. It just completely does it at random. And in fact, what we see is when you look at calico cats, here they are. Look at them, so cute. When you look at their female variation, the reason that they have that beautiful pattern of oranges and black is because their fur color is determined by their X chromosome. And randomly throughout the body, the X chromosome is inactivated, meaning that some cells will inactivate at random the orange, which means that that will show a patch of black hair, whereas other cells will randomly inactivate the black, which will then show the orange fur show through. So it just kind of shows you that there is just like no particular pattern. It's in a very irregular form that the X chromosomes are just randomly inactivated. How about this example? This might kind of make sense of my little rambling that I was just chit-chatting about with the cat. And by the way, you only see it in the females for the calico cat because obviously males only have one X and one Y. So all of their X's will be fully active. They will not be randomly inactivated. So you either have the black or you have the orange. All right, let's look at this example right here. So we have our little mouse, and we see that they are going to be a genetic female. They've inherited two versions of the X chromosome, and we see happening that this is going to either have an X chromosome that will allow for white fur to develop with the lowercase b, or for black fur to develop with our dominant capital B. Okay, now what we see happening is very early on in embryonic development, at random, we're just going to go ahead and we're going to start inactivating one of the X chromosomes in every single one of those somatic cells. So go ahead and follow the arrow, and it says random X inactivation, and notice how in the different cells, they just at random went ahead and created bar bodies. So they condensed down one of the X's, and they basically inactivated that X chromosome. And as a result, what we see is that when we look at the overall pattern of fur on the mouse, we're going to get little patches because, for instance, this cell right here will go ahead and show white fur because the black fur allele has been inactivated. On the other hand, this cell right here, the white fur has been activated. So in that particular patch of tissue, the black fur will show through. And notice how it's very irregular in the pattern, and that is then how we get our different patches of black and white. It all depends on which X was inactivated and where that X is going to be located in the body, if it's going to be the white or the black fur that shows through. Same thing with the cat. Is it going to be the orange or the black fur that shows through? Now, our X chromosome inactivation, we've also seen that there are some clinical applications to how many bar bodies will be utilized, because we do have some diseases where you have an abnormal amount of sex chromosomes. One of them, for instance, is Klinefelter syndrome. In Klinefelter syndrome, you have, from a genetic aspect, an individual who has 47 chromosomes, not 46, 47. And they have 47 because it turns out that they've inherited 3 sex chromosomes. So right here on the PowerPoint, you can see Klinefelter syndrome is when you have an individual who, when it comes to their sex chromosome, has inherited 2 X's and 1 Y. Now, because they have inherited the Y, that means from a genetic aspect, they are considered male. However, since they have 2 X's, we have to make sure that we do our dosage compensation. And what we see happening is that one of the X chromosomes will be inactivated. So you will see 1 bar body if you were to go ahead and collect your samples and do a histology stain. In Turner syndrome, you have an individual who's being born with 45 chromosomes, and that's because this individual only inherits 1 sex chromosome, only an X. So genetically, she's female. The O means nothing's there, so she's only inherited 1 sex chromosome, and because she only has 1, there's no need to do X chromosome inactivation, so you will not need to inactivate an X. So there will be no bar bodies that would show up on her particular slides. How about this one? Triple X syndrome. So now you have an individual that has, once again, just like Klinefelter's, 47 chromosomes because they inherit 3 sex chromosomes. And in this case, all 3 of them are X's. So that means that they have 2 of them that need to be inactivated. And that is then why on a histology slide, they would have 2 bar bodies that would come into play. And the question now is, what exactly is the mechanism or the technique that's being utilized to actually shut down these X chromosomes and control the dosage representation? All right, so for that, we're going to go ahead and take a look at a particular section of the X chromosome that's going to be called the ZIC, or the X inactivation center, and that one is going to have a gene called the X inactivation specific transcript gene. And this gene is what's going to need to be activated if we want the chromosome to condense down into our bar body and shut down its expression of genes. The entire inactivation step is going to take 3 phases. We're going to have initiation, where at randomly we're going to select one of the X chromosomes in the somatic cells. Then we're going to have the spreading, which is going to be the expression of the X inactivation specific transcription gene. the production of the protein that we're going to rely on to condense down the chromosome. And then finally will be this third step, which is maintenance. Maintenance is going to be important because that bar body or that inactivated X gene, X chromosome, excuse me, will go ahead and continue to be condensed as the cell undergo mitosis and is replicated to produce multiple daughter cells along the way of growth and development. Here is a little illustration of our X inactivation center. So there's our particular region on our X chromosome, and in this case what we're after is we're looking for the gene that's going to help us compact. So that's going to be our excess RNA production, our X inactivation specific transcript gene, and what we see happening is that its initiation is what's going to go ahead and cause the step to occur for the bar body to form. So on this little step right here, we can see the initiation, and the initiation, as I mentioned to you before, is going to happen very early on in embryonic development. At random, one of the X's will be picked in all our somatic cells. Once the X is picked, you're going to see the activation of the sync region and the expression of your X inactivated specific transcript gene. This will then initiate the second step, which is our spreading. And with the spreading, we then see that the RNA is going to start coating the chromosome, and you're going to have the expression of the protein that's going to allow our X chromosome to become severely condensed. And the condensing part is what they call the spreading. After this has occurred, that means that the only gene expression will come from the still active X chromosome. The bar body is in essence fully shut down. And now what we see is that our third step is all about maintenance. And that basically means that as the development continues on and the growth and maintenance continues, every single time that the cell replicates itself, the bar body will stick to or will be maintained in the randomly inactivated X chromosome, and it will maintain or stay intact as it goes from one generation into the next of the cells. So as more and more daughter cells are being produced. I do want to point out that there have been cases where some genes can escape inactivation. There have been cases where the cell will go ahead and express both of the genotypes of the X chromosomes. There's also been cases where maintenance is not very successful, and we see that later on in embryonic development, you can either have the expression of both X chromosomes or that the bar body will go ahead and switch over to the other X chromosome. So there have been known to be some variations, welcome to science, but overall what we see happening is usually the one that's picked at random tends to be the one that will stay condensed and maintain its bar body for the remainder of the organism's life. Alrighty, now I do also want to go ahead and point out what we like to call pseudo-autosomal genes. Pseudo means appearing as if, so some of the X and the Y chromosomes have genes that occur in both of the sexes, in both in the X and the Y, and this will give the appearance that they are inherited from the same chromosome, but they're not. So that's what we like to call them, a pseudo-autosomal gene. So for instance, on the illustration right here, you can see my little X and my Y, and you can see that what they've done for you is they've indicated some of these pseudo-autosomal genes in color code. So we have the red that's the same, we have the yellow section that's the same, and we have the green that's the same. And it turns out that these particular patterns are important because it's going to allow them for to properly pair up the sex chromosomes during meiosis. But keep in mind that these are only pseudo-autosomal genes because the X and the Y are two completely separate chromosomes. In fact, the genes that are unique to the X are called X-linked genes, And the genes that are specific to the Y are going to be called Y-linked genes. And we definitely know that these X-linked genes are different when it comes to autosomal genes because of the fact that when you take a look at your sexes, not all sexes inherit the same number of X's. So what I mean with that is that fathers have an X and a Y, which means that they will always give their X to their daughters and they will always give their Ys to their sons. Whereas when we look at mothers, because they have X's, the mothers will always give off their X's. And that means that when it comes to the male offspring, the son will always inherit the X from its mom and the Y from the father. And this is why there's a 50% chance of you inheriting or of you producing a male or a female, because of the fact that that can either contribute the X or the Y. So oftentimes what you'll hear saying when it comes to X-linked genes is that a male is hemizygous. Not homozygous or heterozygous. Hemizygous just indicates the fact that he really doesn't have an option. The one X that he inherits is his one and only copy. So when you take a look at X-linked genes, unfortunately, if there's a mutation that is X-linked, it often tends to affect the male more than it tends to affect the female, because males inherit one copy, whereas females inherit two. So for instance, color blindness is an example of an X-linked inheritance, right? So let me put a little Punnett square right here for you. So bear with me, as my technique of drawing is also not the best. So here is mom with her two X's, and here is dad. All right, so let's say that mom is a carrier for color blindness, okay? So what we see happening is color blindness is an example of a recessive trait, meaning that you need to inherit both, if you have two copies, both copies have to be mutated for you to have the disease. So if mom has one bad copy, one mutated copy on her X chromosome, but the other X is normal, mom is what we call a carrier. She carries the mutation, but she can still see all the colors because she has that backup X chromosome that's fully functioning. Dad, on the other hand, is not that lucky, right? If dad inherits a mutated X chromosome, since that's its only option, dad will automatically be colorblind, okay? So let's do a scenario where you have a mom who is a carrier for color blindness and a father who, from a genetic aspect when it comes to colors, can see everything. He's completely what we call wild type. He's good to go. So here you have mom and the little dot above her X. This is the one that carries the mutation. And as you can see, one is mutated and the other one's normal. And then dad is good to go, right? Okay, so this is what's going to happen. If that sperm carries an X and it comes in contact with the mutated X of mom, child number one in my little Punnett square is a female. Is this female colorblind? No. She is a carrier because she has that one mutated, but because she has one normal, she can see all the colors. We're good to go. Other option, dad's X comes into contact with mom's wild type or completely normal X, and we now have a female that is not a carrier and also doesn't have the disease. So she's good to go. What's going to happen to the boys? Well, we have two options. Mom, if she gives her mutated version of the X chromosome, dad can only give his Y. So guess what happens? This little boy right here, colorblind. The other option right here, obviously, is that mom gives her wild type, completely healthy X chromosome. Dad gives his Y, and the child is good to go. There's absolutely no abnormalities, no carriers at all. So you want to kind of think about this when you're looking at X-linked diseases, because oftentimes what we see, and we see this, like I said, in color blindness, we also see this in hemophilia. Not all hemophilias, but most hemophilias, they'll have an X-linked factor to it, meaning that the mothers are often the carriers, meaning they have one mutated version and one good version, and they have no idea that they're carrying the disease because they're showing none of the symptoms. And they tend to go ahead and they give the disease to their sons because the sons inherit the X chromosomes from mom. So here, for instance, we have an example with our X-linked gene, and we're taking a look at muscular dystrophy. Muscular dystrophy is also studied in dogs. We see it, and I'm going to show you a picture of a little dog with MD in a little bit. But basically what we see happening in this mutative disease is that the little dystrophin protein, which is kind of like an anchoring protein that allows you to take the contractile force inside the muscle cell and communicate it to the sarcolemma or the membrane on the outside of the muscle, that anchoring protein is not functioning. So they're not able to contract and relax their muscles. So even though the brain is sending signals, the muscles are not responding. And it can usually cause a lot of damage, especially to the heart and the breathing muscles. It can be a very painful disease, and unfortunately, even with the best treatment, usually the survival rate is not looking all that well. They tend not to have a very long life. Now, we also know that for this particular disease, it's carried on the X chromosome. And look what it says right here. It says it's X-linked recessive. So that means that the only way that a female can get it is if both of her copies are mutated. If only one copy is mutated, then she is what we like to call a carrier. So she has none of the symptoms, but she carries the mutation. And the problem with this is then that when it comes time for you to do your little Punnett square, like we did on the previous channel, you have a 50% chance that if you're a carrier, that you will give one of your mutated X's to your sons. And if that happens, your son has the full-blown disease. And we see this happening over here on our little pedigree. So you can see that the male, absolutely no traces of the disease, right? And we can see the female right here, it's half because she is heterozygote. So she only carries one mutated copy. She's unaffected by it because she is the carrier. And what we see happening when it's time for them to produce their offspring, only the sons will have the chance to be fully affected by the disease. The daughters will be the carriers. Now, in case you're wondering, is it possible for the daughters to have muscular dystrophy, to be colorblind, to have hemophilia, any type of disease that's X-linked recessive? Yes, but in order for the daughters to be affected, that would mean that they would need to inherit both a mutated copy from mom and a mutated copy from dad. And since that mutated copy also has to come from dad, that would mean that if we're looking at our pedigree, guess what? Dad would be indicated that he has the disease. Because remember, dad only has one X. So if that X is mutated, then dad would have the disease. So if we were to redo this pedigree with dad having the disease and mom being a carrier, then what we would see happening if we put that in a Punnett square, that scenario would allow both the male and the female offspring to carry the disease. But if there's a scenario such as the original one right here, where dad has absolutely no traces of the disease and mom is just the carrier, then in that scenario it will only affect the male offspring, and the females will be the carrier or have the potential of being the carrier. Now, oftentimes when they're trying to study X-linked diseases, they're going to do this thing called a reciprocal cross. A reciprocal cross is when you do two crosses that will differ in which sex will carry the particular trait that you're looking for. So for instance, go ahead and take a look at the two Punnett squares. And what we're looking at over here is on our first scenario. We switch back over to my pen. We see that we have an unaffected female and we have an affected male. So see how you have the lowercase d that indicates that they have the mutation. So in this case, the male has the mutation. Mom is completely healthy. No problem at all. And what we see happening is since dad is the only one that's carrying the mutation, that means that you have a chance that he can go ahead and give his X, and it will combine with the healthy X of mom, and you have a carrier daughter. but it could also mean that when it comes to look at the boys, look what's happening. None of the boys are affected. And the reason for that is because mom's X's are completely healthy. And remember, when it comes to the male offspring, all the X's come from mom and all the Y's come from dad. So you have a high chance that the daughters are carriers because dad's X is mutated, but because mom's X is normal, the daughters are carriers. They do not have the disease, and it doesn't affect the boys at all. Now, here's the reciprocal cross. So we're going to take the same scenario, and we're going to switch our sexes. And what we see happening is that now we have mom that is carrying 2 of the mutations. So she has a full-blown disease, and dad has absolutely no mutations. So notice how this X has the mutation and this X has the mutation. And that means that if mom is walking in with both of her X's being mutated, not only does mom have the disease, it also means that all of her daughters will be carriers because all her X's are mutated and dad's X's are normal, wild type, and all the sons will have the full-blown disease because remember, all the sons inherit their X's from mom, and in this case, both of mom X's are mutated. So in a reciprocal cross, we tend to study X-linked diseases, and basically what we're doing is we're doing a scenario where you have the disease differ in which sex carries the trait. So it allows you to show how the gene will behave differently, and it will allow you to trace the carriers as well as the individuals that are affected with the disease. All right, guys, let's stop this particular part right here. This is towards the end of chapter 4. So when we do our next audio lecture, we'll pick it up from chapter 5. I also want to take this opportunity to just kind of remind you that if you have any questions, comments, or concerns, please feel free to email me, drop them in the discussion session, and if you guys want to do a Blackboard Collaborate, if I'm not already in the room, just send me a quick email and I'll hop onto the computer and we'll get it done, okay? So don't be shy. Let me know if you have any questions, comments, or concerns.